My name is Abigail LaCour. I'm an undergraduate researcher from Thomas Jefferson University in Philadelphia, Pennsylvania. I will be presenting our research on the natural dye potential of invasive plant species for commercial textile applications. To summarize, our research aims to identify sustainable alternatives to synthetic dyes in the textile industry. By investigating invasive species as natural dyes, we are providing an innovative opportunity for successful environmental rehabilitation. Our previous research showed that the roots of wineberry, bittersweet, and barberry show potential as dyes, while our current research explores the efficacy of the berries as dyes. Invasive species negatively impact the ecosystem because of their ability to reproduce and grow faster than native species. Our current species of focus is Rubus phoeniculaceus, or Japanese wineberry. Its berries contain high levels of anthocyanins, which provide ecological functions to the plant, including antioxidative, antimicrobial, and photoprotective effects. The red to violet coloration serves to attract herbivores for seed dispersal, but may also be effective as a natural dye. Figure 2 shows the geographic range of the invasiveness of wineberry in the United States. The prevalence of invasive shrubs is increasing in the United States, and providing commercial use would hinder their replication and therefore their impact. Synthetic dyes produce effluents containing metal neurotoxins which bioaccumulate. Natural dyes are non-toxic alternatives. Currently, only 1% of textiles are dyed with biomaterials, with expected growth to 5% by 2027. Hues and dye efficacy depend on chemical composition, which has directed our research towards chemical analysis. Below are figures showing the textile colors in demand. As an overview, we extracted dye from berries using two extraction methods, cold and hot. Samples were centrifuged and their composition was analyzed by spectroscopy. Berry extracts were dehydrated and masses were used to determine crude dye yield. For cold extraction, berries were combined with 3 solvents, water, vinegar, and alcohol. Solutions were stored at 3 degrees Celsius for 7 days. For hot extraction, pulverized berries were transferred to a thimble and transferred to a soxhlet apparatus with DI water as the solvent. The samples were heated for 100 degrees Celsius for 6 soxhlet cycles. Prior to analysis, samples were centrifuged to remove suspended plant particles. The absorbance of each extract solution was measured using UV visible spectroscopy. Extraction solutions were set to evaporate at room temperature for 7 days. Dried samples were collected and weighed to determine crude dye yield using the equation below Figure 11. Figure 12 shows the absorbance spectra of the extraction solutions ranging from 512 to 534 nanometers. Figure 13 shows the crude dye yields from 4 wineberry extracts. The vinegar and hot soxhlet extractions produced the highest yields. Studies that used UV-Vis to analyze the anthocyanin content of berries found maximum absorbances at 520 nanometers within the range of our findings. Higher yields are associated with stronger bonding to of the solvent to anthocyanins, and prolonged extraction is expected to increase dye yields. In conclusion, wineberry shows promise as a natural textile dye. Anthocyanin content was verified through UV-Vis spectroscopy. The next steps in our research are to evaluate and quantify the anthocyanin content of barberry and wineberry berries. Following chemical analysis, we will test the efficacy of the berry dyes on wool and cotton using 3 mordants while following AATCC industrial standards.